Rolling up.
Alright. Set.
Action.
We can use this imaging system which has a UV light in it. We've stained our samples, which will stain the DNA and enable us to visualise whether the DNA is at a size that we predict using what we call a ladder, which has DNA fragments of known sizes. What do you want me to do?
It's got enough paste under it.
Just press some more buttons. It's good.
Oh, okay.
What's happening now, Lauren?
I'm just looking at some of the images that we've acquired. Past images.
I think so.
Is that
Set. As you were saying.
So I've got some cells in here. I've manipulated these with CRISPR. So CRISPR is a gene editing tool, so it allows us to be able to edit the DNA within the cells of interest that we're using. And CRISPR has been originally was developed from the bacterial immune system, but the way that we use it and what's really important is how we use it in gene editing. It allows us to make specific changes to genes that we may see in cancer, things like make point mutations. We can, which is changing a single base within a gene. So in terms of the genetic code, you'll see A, T's, G's and C's that make up all of our genes within our genome. And what happens in cancer is that there are changes that happen to that genetic code that are not normal, and they turn normal cells into cancer cells. And so we can use CRISPR as a way to be able to manipulate the DNA of cells that we use in the lab to understand how specific changes, whether that's specific sequence changes within the DNA, so changing one of those A's to a G, for example, whether we want to delete a gene. So is that gene required, essential for the cancer? We can delete it. In the same way, we can alternatively overexpress it. So that just means expressing it at really high levels within the cell, so turning the gene on. So there's various sort of manipulations that we can do with a CRISPR system. In terms of how we practically use it and generate cell lines to be able to edit the DNA, there are two components to the CRISPR system. One is a Cas9 or Cas13, which can edit either DNA or RNA. But essentially that is just an enzyme, an endonuclease that can cut DNA. It acts like a pair of scissors, which enables you to edit. And then the second component of it is what's called a guide RNA. It matches the sequence that you want to target within the genome. So that is a very specific way for you to be able to direct your editing system to specifically where you want to target in the cells.
Beautifully said. Let's wait about halfway through the very technical for me.
Yeah.
What's the point of editing? And that we can express it and turn it on, you can turn it off.
So the point of editing is for us to understand, in our context, is for us to understand what changes we've identified in patients, for example, with childhood cancer, which is our area of research interest, how those changes actually affect the behaviour of a cell.
Can you give us an example of what you've done within your research that this applies to?
Yeah, so we may be looking at genes that are potentially drug targets, for example. So we may have a gene that is expressed really, really highly in a patient with a childhood cancer, whatever, say that's a sarcoma or a brain tumor, and we know that that's abnormal. It's not usually expressed that highly in normal populations, in normal cells. And we have a drug that can potentially target that specific gene. However, what we need to know is whether that gene is actually what we call driving the cancer. Is it essential for that cancer cell to keep proliferating and surviving? And so the way that we can do that is we can use CRISPR. We can specifically target that gene, so delete it, so take it completely out, and see whether that kills the cell. If that actually kills the cell, we then know that that is what we call essential for driving the cancer. And then we can take that back to a patient context and infer from that that a drug that also targets that specific gene would potentially be beneficial for the patient.
So you're removing alternatives until there's none left.
Yeah, sort of, yes. And we can either use
If you
CRISPR.
could say that,
Oh, okay, yeah. Yeah, so it's a way of refining drug targets or honing down on what may be a good target. And we can use CRISPR in either a very targeted way where we target a specific gene, or we can also use what's called a CRISPR screen, which is when we target all of the genes within a genome in a very high throughput manner, and that's kind of beyond what we're doing here today. But that will then tell us unbiasedly when we target this specific gene, that will kill the cells.
one of the quotes we've been bandying around since we started this is from Sherlock Holmes.
Right.
And he says, once you've eliminated all the possibilities of impossible, whatever you're left with is the truth.
Yep. Yeah, I mean, I guess that would apply here. So yeah, everything that we do is about understanding how linking the genetic changes that we see in patients to drugs, and the way that one of the tools that we use to be able to understand what those specific changes do to a cell. Are they actually driving the cancer, and therefore would it make an effective therapeutic target, is by using CRISPR to be able to manipulate cells within the lab to see what those genetic changes do.
How long has CRISPR been around? What are you talking?
Oh, gosh, I don't know, 20 years, longer? Look, I mean, definitely my whole career. But we've been using it very routinely, I would say, for the last 10 years, the last decade. Yeah. So it's a very sort of core technique that we do use in the lab.
So it was a breakthrough, whoever discovered it.
Yeah.
How the hell did you discover it? I know you won't know.
I don't know. I think it would be, one of these kind of incidental findings that we often see.
What's the basis of it? You can get right in and literally cut.
Yeah. So and that's the function of the protein, what's called an endonuclease, is to cut DNA. And then we've been able to manipulate this system to be able to guide it to specific areas of the genome to do what we want to do.
Exciting when you see it happen?
Yeah, yeah. Well, I mean, we obviously don't see it at like a microscopic level, but in terms of like how we use it, it is incredibly useful for understanding the function of genes in cancer and specific genetic changes in cancer.
Before CRISPR, you couldn't tell what it was doing.
I mean, we used, and we still use these models instead, rather than specifically editing the DNA within the cells themselves. We a method that's called overexpression. So we insert our gene of interest, whether that's a kind of normal gene that we just want to express at really high levels, or a gene that's been mutated. either one of those, single letter changes, an A to a T, or part of the gene's deleted, or gene A is fused to gene B. There's all sorts of rearrangements that occur in cancer. But all we kind of could do, the way that we were able to model that, was just to insert that mutated gene into the genome of our cells of interest at a really, really high level and see what it did.
How long has the genome been around?
20
20 years?
years, yeah.
So it's post that.
Yeah. Post that, we could edit these cells in this way before the whole human genome was sequenced. So it's a very, very old technique in the traditional way that we do it. But when we do it in that way, where we just overexpress it and leave all of the normal genes of the cell that we're working with as is, there's no way for us to control what the impact of it is on that gene that's still in the cell.
I love the way you say it's very, very old and you're referring to 20 years.
No, it's older than 20 years in terms of that method. Our ability to be able to, overexpress genes that, as they're kind of discovered in these types of systems, predates the sequencing of the whole human genome.
And it's still such a useful tool.
We still use it in parallel with CRISPR. So if we're talking about, so this is what this looks like. In terms of what we use CRISPR for in the lab, and when we're trying to really understand what genetic changes do to cancer cells, we try to do this by as many techniques as possible to be able to understand. So if we're talking about overexpress or high expression of a gene, abnormally high expression of the gene, it's really great if we can show with CRISPR that knocking that gene out or deleting that gene, that kills the cells. So that tells us that it's kind of essential. But also, on the flip side, if we use the old ways that we used to do it and we just express that gene really highly in a cell, does that, on the flip side, make that cell grow really quickly, replicate really fast, which would be the kind of features or hallmarks of cancer. So kind of complementary approaches.
Two things quickly before I let you get back.
Yeah.
What quickly are its virtues? CRISPR's virtues, coming from nothing to that. And what can you see that might be ahead that will replace CRISPR, if anything? And you might not know, I have no idea.
In terms of its virtues,
Well, no, the two separate questions.
Yeah.
What are its inherently? What are CRISPR's virtues?
In what makes it beneficial, like from a practical sense?
Absolutely.
I think it's how we can adapt it, how we can tailor it now. We used to only use it for deleting genes, pretty much. And now we can use it for so many other applications. We can use it to, as I said, introduce single point mutations. We can use it to introduce partial gene deletions. So rather than deleting the entire gene, we can just delete a small region. We can chop two genes and then make them fuse together, which is something, so causing a fusion gene, which is something we commonly see in pediatric cancers. We can turn a gene on. There's lots of different things that we can do with CRISPR now that we couldn't do, 10 years ago.
Is it because nobody thought about it? Was it just underutilized, or somebody said, "Oh, we could use CRISPR to do that"? Or was it?
I think it's just how these things develop, right? Thinking more about what further applications could be. The people that have managed to manipulate these systems are far smarter than I. So how they actually, were able to understand that they could use it that way, that's kind of beyond me.
So it's lateral thinking. That would be good for this.
Yeah, absolutely. I imagine so, yeah.
No idea what's going to replace it, or it'll always be with us now.
Yeah, I'm not sure. I mean, as I said, it's incredibly core to what we do. It's a core technique that obviously us in cancer research, but in many other fields are using it as well.
Thank you. No, you were saying.
Okay, one more you're right. And just do that again if you would. And let it go black.
Here we're looking at some cells that we've manipulated with CRISPR. We want to make sure that we've got our two components of the CRISPR system expressed in the cell. The way that we do this is by tagging them with fluorescent markers that we can analyse using this machine, which is called a flow cytometer. And so we have our Cas9 endonuclease, which is the first component, which is tagged with a red colour that we can see along this axis here. And then our second component is called a guide RNA, which matches the sequence within the genome that we want to target, and that's coloured with a green marker, which we can see along this axis here. So for the cells that we want to analyse, we want to make sure that they have both of these components, so they will be both red and green, and they'll fall into this top quadrant here.